Hi everybody, welcome to our first biology skill builder. Today we're going to be talking about an introduction to the scientific method. The scientific method is a process used by scientists to study the world around them. It is a logical, systematic approach to solving problems and finding solutions. So I'll start with a quick overview of what this looks like. The scientific method starts with identifying a problem and forming a question that can be tested. So a scientific question can be answered by making observations and gathering evidence. The second step in the scientific method is to form a hypothesis. You may have heard this term before. A hypothesis is a possible explanation for a set of observations, and it also serves as a starting point for further investigation. Following is the next step in the scientific method, and that is to test our hypothesis by designing an experiment. We will spend some time today learning about a control versus experimental group, as well as the differences between an independent, dependent, and controlled variable. The next step involves the collection of data. So data consists of facts, figures, and other evidence gathered through observations. In this lesson, we will actually compare different types of data, including qualitative data versus quantitative data. Following is data analysis, and the final step is to communicate the results to our scientific community. The scientific method starts with identifying a problem and forming a question that can be tested. A scientific question can be formed by making observations with your five senses and gathering evidence to form an inference. Before we get started, let's go ahead and clarify the difference between an observation and an inference. Observations are made using your five senses and include things that you can actually see, whereas inferences are things that you suppose are likely to happen based on what you see. Let's clarify this a bit further. In the following text, identify which statements are observations and which ones are inferences. I anxiously waited outside of Miss Lai's class for Chloe. She walked out with a huge smile on her face. She didn't even get the chance to say a single word before I ran over to her and embraced her. We jumped up and down ecstatically. I knew she must have passed her final. Our parents had to let us go on the school trip now. Go ahead and pause this video and take some time to identify which statements are observations and which ones are inferences. The statements, she walked out with a huge smile on her face, and she jumped up and down are observations. There are things that you can actually see. However, the statement, I knew she must have passed her final, would be an inference. Because the speaker saw Chloe smile and jump, the speaker supposed it was likely that Chloe passed her final. Let's make some observations using your senses now. In this image, we can see that Cactus B is shorter than Cactus A. We can also see that Cactus A is sitting closer to the window. We can also see that direct sunlight is able to pass through the window. Now, let's make an inference. Based off of those observations we just discussed, one could suppose that there may be a relationship between the amount of sunlight exposure and plant growth. Now, we can form our scientific question. The question you ask needs to be something that you can measure so that the results can be compared. For example, how does the amount of sunlight exposure affect plant growth? That would be a testable scientific question for this scenario. It is also important to do background research to find out what's already written about your question before you start an experiment. The second step in the scientific method is to form a hypothesis. A hypothesis is a proposed explanation for a set of observations and serves as a starting point for further investigation. A hypothesis must be testable and measurable. A hypothesis is written as an if-then statement. So let's think about what a hypothesis would look like for our experiment. If the amount of sunlight exposure to a given plant increases, then plant growth will increase, would be a simple hypothesis about how plants grow. In this example, plant growth can be measured. The next step in the scientific method is to test the hypothesis by designing an experiment. This includes a list of materials and a procedure or step-by-step explanation of how to conduct the experiment. Scientists must be careful in how they design an experiment to make sure it tests exactly what the hypothesis states. A proper controlled experiment compares two or more groups, but changes only one variable. The control group is the group that is used for comparison. It does not receive treatment by the researchers or the variable being tested. For example, the group of plants that receive no sunlight would be considered the control group. Experimental groups are the ones that are compared to the control group. Experimental groups do receive treatment by the researchers or the variable being tested. For example, when testing the effects of amount of sunlight exposure on plant growth, the groups of plants that receive specific amounts of sunlight exposure would be the ones considered to be experimental groups. We just used the word variable, but let's take the time to define what that means and look at different types of variables. Variables are factors that change in an experiment. Independent variables are those that are tested and changed by the scientist. In our experiment, the independent variable is the amount of sunlight exposed to each plant. The amount of sunlight is tested and purposefully manipulated by the scientist. Dependent variables are measured by the scientist and changes as a result of the independent variable. So in our scenario, the amount of growth that occurs in each plant is the dependent variable. Plant growth responds to or is a result of the amount of light it was exposed to. Controlled variables are variables that are kept the same or constant in all setups. This is important in making sure that your experiment tests exactly what the hypothesis states. Otherwise, an experiment could result in faulty data due to confounding or external variables. In our experiment, some controlled variables that we would think about include the amount of water given to each plant, the type of plant used, and the type of soil used. If plants received a different amount of water, it would compromise the results, and our study on the effect of sunlight on plant growth would not be accurate. Keeping detailed, accurate records is an important part of the scientific method. Data includes the facts, figures, and other evidence gathered through observations. There are two different types of data that can be collected when performing an experiment: qualitative data versus quantitative data. Qualitative data is the data type that consists of descriptive statements. They are text-based and include descriptive words such as color, texture, smell, taste, and appearance. On the other hand, quantitative data is the data type that can be measured and expressed numerically. This type of data is number-based and includes things like length, height, area, volume, weight, speed, time, and temperature. If you are wanting to perform an experiment with results that could be statistically analyzed, then the collection of quantitative data would be important. Oftentimes, a visual tool such as a chart, table, or graph is used to represent data collected. All visual representations should be clearly labeled, concise, and well organized. The title, axis labels, and key should all be identified. A data table provides you with an organized way to collect and record your observations. For example, in our data table, we listed the independent variable, in this case the amount of sunlight exposure, in one column, and the dependent variable, plant growth, in a separate column. From this table, we can also identify that plant A is the control group because it receives no sunlight and is used as a comparative group. On the other hand, plant B and C are known as experimental groups because they receive specific amounts of sunlight. Then you can use your table to create a graph. Graphs help you understand and use the data. Graphs make it easy to identify trends and make predictions. The x-axis or the horizontal axis of your graph represents the independent variable, while the y-axis or vertical axis of your graph represents the dependent variable. Note that the x-axis is labeled amount of sunlight exposure in hours, and the y-axis is labeled plant growth in centimeters. In our next lesson, we will spend time looking at different types of data representation and look at how to interpret and analyze the data in more detail. The next step in the scientific method is to analyze the data. Data analysis is the process of interpreting the meaning of the data we have collected and organized. This process involves looking at patterns, or similarities, differences, trends, and other relationships, and thinking about what these patterns might mean. We will spend some extra time analyzing data in our next lesson. The scientist then summarizes their findings and relates them to their hypothesis. Was the hypothesis supported? If not, a new hypothesis is developed, a new experiment is conducted, and the new data is observed and analyzed. The last step of the scientific method is to communicate the results. After you have gathered and analyzed your data, draw a conclusion about your hypothesis. A conclusion is a summary of what you have learned from an experiment. It is then important to communicate your results to the scientific community. By communicating research results with colleagues, we allow ourselves to receive credit for doing the work, connect with others doing similar work, thereby leading to new collaborations, and establish our results within the scientific knowledge that future research will be built upon it. There are several ways that scientists communicate results, including written and scientific journal publications, by giving presentations to our colleagues and the public, or scientific conferences. Today we looked at the 6 steps of the scientific method. Number 1, identify the problem. Number 2, create a hypothesis. Number 3, design an experiment. Number 4, collect the data. Number 5, analyze the data. And number 6, communicate the results. Thank you guys so much for joining me today in our introduction to the scientific method. I'll see you guys in the next video. Bye!